And that's in the line of the main subject of this symposium. It's on trinity of drug discovery. So we'll be talking about targets, about compounds and inhibitors that were discovered by our colleagues and our company in collaboration with different organizations. And certainly it would open an avenue to different chemical spaces. Sorry. Yeah, and this protein kinase CK2 is present in really in many cellular compartments, and it is able to phosphorylate, I think, hundreds of substrates, and for this reason it was linked to several cellular processes and transcriptional control, neural function, response to cellular stress, and also CK2 is involved in many pathologies, in particular it's cardiovascular diseases and atherosclerosis, cardiomyocyte hypertrophy, neurodegeneration disorders. But right, really that is one of the targets, and we started exploring CK2 about 15 years ago, and which is at that time it was not really a popular target, but it was kind of scientific interest, and that is also a good model for our to test our approaches in silico drug discovery. So that was again a combination of targets, and we in the company we collected a very nice collection of compounds, and trying to go through that collection to look for new inhibitors, and at that point CK2 was chosen as a target, and so obviously it was an X-ray data for that kinase with different compounds, and certainly its studies really demonstrated that it's really a abnormally elevated CK2 level is required for tumor progression, and dependency of cancer cell to high level of CK2 that comparing to normal cell provides really crucial argument for development of selective CK2 inhibitors in cancer therapy. Over the years, a number of organizations and research groups worked trying to work on this target, and discovering really huge array of different classes and families of CK2 inhibitors. And really that is important. Just a little bit of history in this respect. Those compounds were found by, I would say, try and error approach. So that is kind of many of them were discovered, I wouldn't say by accidentally, but there are no logic behind of discovering those scaffolds or targeting those molecules. And with the development of in-silico screening technique, really we could focus on discovering new scaffolds and finding new inhibitors. And also we are talking about selectivity. We know better about binding process of those inhibitors. And that is just, generally speaking, is representative examples of different classes. And certainly there are coumarin, flavones, anthraquinones, parazola triazine compounds, and we'll be talking about these compounds CX4945 that is in clinical trials right now. So that is like really interesting compound, and it has kind of long history for this material. One of the primary reason it was in the development, as far as I remember, from 2009, this compound, and this is first in class, like really potent and highly selective inhibitor for CK2, and this particular compound really demonstrated favorable safety, pharmacokinetic characteristic, pharmacodynamics, and it's in clinical studies, in phase 2 clinical studies. And as you can see, structure is pretty simple, but as I said, it's in phase 2 clinical trials compound. But what is really interesting is that Senhua Biosciences, that company in Taiwan, is now in charge of clinical trials for this material. And what is it's in trend, what's going on right now. It touches oncology, obviously, and it's the main purpose of use of these materials is oncology. And what is important to say is that they found that it could also could be also promising therapeutic agent due to dual impact on COVID-19, which is, and in this respect, it's very interesting to say that might be among a pool of CK2 kinase inhibitors, it would be, I would say, a drug which is maybe even more potent than this compound, and certainly additional studies are necessary. And that is my, again, in this respect, my personal opinion, that we are very focused on vaccine development and really drug discovery in that direction, at least to what I see in the news, etc., is kind of hidden from the public. But treatment in this respect and finding new drugs and repurposing drugs maybe is important approach to target COVID-19 pandemic. And it's interesting that this CK2 inhibitor compound, it's entered already in clinical trials for treating COVID-19, and there are first very interesting and encouraging results for that. And coming back to our approaches, and as they mentioned, for in-silico drug discovery, which helps us to rationalize what compounds to select for initial studies, and then it's a guidance to discover new chemical spaces and new compounds around those materials. And obviously, in our company, we are focused on two different approaches, and it's receptor-based target libraries discoveries and ligand-based target library discoveries. And so here it's standard approach, I would say, in the modern drug discovery world. But what is important to say is that crucial improvements at every stage are really necessary. So, for example, if we are talking about receptor-based approach, so we focus on ligand preprocessing, and our company, and with collaboration with a number of organizations, we did crucial improvements with this, how we process ligands and how the programs could see them. And obviously, once we have a number of hit compounds from in-silico screening, we are doing visual inspections that requires its human-involved protocol, but it is really important part of the process to have the targeted library and for next in vitro screening. Again, it's a standard legal ligand-based approach that is also a part of our drug discovery process. And in a minute, I'll start talking about like really compounds our company is working on with a number of research institutions around the world. And ligand and combination of these two approaches guide us to find new scaffolds, and based on those new scaffolds, we can go into new chemical spaces and find new inhibitors, which I'll show you in a second, like really shows good profiles. And that's just certainly, just I have to mention that over the years we discovered about over 25 different scaffolds of CK2 inhibitors. And really, I'm not going to talk about all of them, but to show you a little bit of evolution of the approach and how in silico data combined with access to compound databases or available compound databases and their structural modifications, and allows us to go into new chemical spaces and rationally look for new materials which you wouldn't think for just error and try approach. So certainly in silico, once we have molecular modeling and it shows key interactions of compounds, and then if I come back, I'll focus in the first slide of this PNH79 material. It's a flavon type compound which showed like really great selectivity in our studies on a small panel of protein kinases. So it's really important. So that we did not try the whole panel that is available to test, but as an initial try, it's a good start. And that's again, as I'm saying, it's a molecular docking experiments that shows us key interactions of this material in the protein with the protein and ATP binding site of CK2. And then let's say go into more details on how we rationalize when we are looking for a new compound. So that is a general slide where we discovered two potent CK2 inhibitors that we call it FLC26 and FLC21, and a little bit of history of how it was discovered and our approaches to this. And initially we took the small library, relatively small library of 150,000 compounds doing docking experiments, and ending up with around 2,500 compounds to test. And after visual inspection, as I said, it's also an important part of the drug discovery process in our environment, in our company. We ended up with testing 500 compounds that and some results, and it's the results you could see here. It's for compounds with off the shelf, so that no synthesis required at this time, but already it gives you a good idea on which structures to focus. And we got compounds in with micromolar activities, And that is was like really interesting, and the first step to do to generate even more potent compounds. So first of all, comparing their structures of the most active compounds, we came to a conclusion that the first one here is that you have to have a substituent in position 6. So position of hydroxy group at position 3 is also important, and position of carboxylic group at around the ring B at 4' position it also plays role. So, and we ended up that 2 groups are fixed. It's hydroxy group at position 3 and carboxylic group at position 4'. So based on those results, we could now see the compounds and go into more detailed structure activity studies. And that's the result. A number of materials were tested, and the most interesting and the most potent compounds were selected. So as I said, we call them FLC21 and FLC26, and that's already a compound with nanomolar activities, and that's the story behind of selecting substituents at position 6 and 8. So the first line of studies were to see which which group is or substituent is the best to have at position 6, and we found that halogens are preferred. And the same story was, do we need just one substituent, or it's better to have 2 at position 6 and 8? And so we ended up with compounds to have dichloro or dibromo substituted materials. Again, that's we did a selectivity profile of these 2 compounds, and it's not perfect comparing to their material, the compound I showed before, but we're in the right direction, and only Aurora A kinase was targeted by these 2 materials to a certain extent, but still it shows rather good selectivity over the other compounds, the other targets. It's important to say that molecular dynamic simulations for our experiments, it shows vital interactions of these molecules with amino acids in the protein, and that is important. We could extract important structural information from this data when we are logically planning the next set of experiments, and I'll show that in a minute. That is important part of the interactions of this flavonol compound with key amino acids in the protein, and that is taken from the paper, our molecular dynamic simulations, especially when you don't have X-ray data. That's an important chunk of information you can extract from just computational studies of the interaction of compound interaction with proteins in CK2. And based on this, we have complex of CK2 and one of the compounds FLC21 using molecular docking. And fortunately, we managed to get X-ray analysis of this compound with the protein. So it was a very nice experiment showing that our docking experiment, our docking experiments are really important and giving us valuable information about structural interactions of these of CK2 inhibitor with the proteins. And that is next slide is a superposition of these two complexes. It's molecular docking experiments and X-ray analysis, and it's really in good correspondence with the results obtained just in silico and real-life X-ray analysis. So we are summarizing all this. I would say that we have a pretty good and robust model of predicting and selecting new classes and new scaffolds of potential CK2 inhibitors. And that's led us to discovery of a new class of compounds called aurons as potent inhibitors of human protein kinase CK2. And as you could see, it's a compound that even more potent than VLC26, and keeping in mind all the structural information we got from our previous studies, we managed to rationalize that this class of compounds could be good starting point to look for new scaffolds and obviously to have more selective materials. Here we are showing that comparing distance between key interactions of flavone and auron derivative. So that is interesting observation, and as I said, keeping in mind that we have a pool of information from our previous studies, and based on just structural features of auron derivatives, we could rationalize that this compound could be a good starting point looking for new CK2 inhibitors. And geometry and structural features of those materials confirmed that. That is also important slide in showing that binding mode of VLC26 obtained with with X-ray analysis, and the most potent inhibitor, I'll talk about it a little bit later, that is in orange, obtained with molecular docking. So it was a first starting point for our discovering of new class of inhibitors, and we could rationalize that we could even do better by fine-tuning of structural features of these inhibitors. And that is we also took into consideration a few other parameters for compounds and like lipophilicity and log-P, C-log-P parameters, and try to rationalize in which area of the structural scaffolds we have to look for new compounds. And that, as you see in red dots, we took our own derivatives before structure optimization. But keeping in mind all this data, we rationalize that we could predict and synthesize our own derivatives that would have a better activities compared to compounds we have, I would say, off the shelf. And green triangles indicate our own derivatives after optimization. And we synthesized a number of materials, and just 4 of them you could see at this table, and we call them BFO 181, and 4 the most interesting and active compounds are shown here. So, and if I could just come back, so you could see the structures, the most interesting, the most potent material so far, it's BFO 13. And so that's the story behind flavonoids and aurons shows as we could gradually progress into the direction of finding new materials, new inhibitors, and our approach, how we achieve that. And at the same time, it's important to mention that we are exploring rather small chemical space, which is limited to commercially available materials, and especially we have in our company, in our possession. But it opens an avenue to finding new scaffolds and new materials. And in that respect, that could guide us to discover new chemical spaces and new areas of the chemical spaces. As previous speaker mentioned, it's a really huge universe of the possible materials, and obviously we cannot randomly explore it. We have to have some rationale behind that. And keeping all this in mind, we focused on and at utilizing all our knowledge we have. It's kind of, I would say, became very easy to find new scaffolds. Once you do docking experiments on new libraries and see the compounds which are perfectly binding to the active site of CK2, it was not a big surprise for us that we found immediately new scaffolds and potent CK2 inhibitors. So that's thionopyrimidine compounds. Again, that's an example of molecular docking experiments and set of compounds that we found are active, and certainly structure activity relationship showed us the preference of chemical groups to be in certain places. And based on that, we could rationalize and synthesize new set of compounds and tune their properties, not to the perfection, but to have a good compound again with decent selectivity. And as a result of those studies, we discovered TTP22, and also selectivity profile is shown here. Again, we have a rather small kinase panel to check, but again, it's rather selective, and the only worry it Aurora A us, that the selectivity profile could be tuned even better, but certainly it shows us that just in a couple of steps we could generate new potent and micromolar compounds with micromolar activities. Again, that is general scheme. We took completely different scaffold, and it's thiazole carboxylic acids, and and after screening and structural optimization, we found 3 compounds again that is micromolar with micromolar activities. And I should mention that these compounds again, were discovered just in 2, for us, already simple steps through molecular docking experiments, digesting all that information we have and selecting screen compounds. And then based on discovery structure activity relationships, we could synthesize a new set of compounds, again, do molecular docking experiments, and end up having interesting and promising CK2 inhibitors. Again, that's just a general slide showing that over the most recent discoveries, another scaffold of imidazopyrimidines, and again, those compounds with micromolar activity, and based on structural information we got from our previous experiments, we could rationalize why we have activities of those compounds and how to improve them. So, saying all this, it's we have a pretty robust system of discovering new inhibitors, and as I said, CK2 was a model studies for us, and we took just a separate library of compounds and did molecular docking experiments, pharmacophore screenings, and just based on those results, we took almost 300 compounds that for the biological tests, and immediately we saw 18 compounds that have inhibitory activity against CK2 in micromolar range. And what is very interesting is that we have 15 different chemical classes of among those 18 compounds. That it means it's again very interesting result in terms of opening new chemical spaces. And again, if we are coming back to the topic of this symposium and the beginning of my talk, that all our activities are in the direction of finding new scaffolds and opening new chemical spaces. So for specific, in this case, for a specific target, but that could be true for any other target where we will be talking about and people will be talking about, that you have to have a pool of compounds to work with to find that perfect material and perfect drug candidate, which eventually would lead to at least phase 1 clinical studies. But again, that is done on a pool of known commercial materials. And then structure optimization, we have inhibitors, but that is...
You have 10 minutes left.
Sure, thank you. Yeah, and just a couple of slides. That is Greg Benford in his book Shipstar made very nice point. When the chemistry is right, all the experiments work. So in this respect, we are very pleased to announce that we are working on Cambria. It's a new multi-billion-sized pool of chemicals that is should be available within a week or so to general public and for therapeutic research and development. And it's a chemical space comprising of over 11 billion tangible screening compounds. And that is based, that chemical space we are compiling is based on chemical reactions which are mastered in our laboratories and based on chemical building blocks that our company has. So again, if we are coming back to that expression on the slide before, we do the right chemistry, and now we should make all experiments work. So immediately I tried, when we compiled that space, I took that compound CK2 inhibitor, which is in clinical phase 2 clinical trials, silmetasertib, and run it against the chemical space we are producing at Cambria. That's this Cambria chemical space. And as you could see, it's very interesting. The similarity is 0.98. And I took the liberty of checking again other chemical spaces available, and that's the best similarity result we could get so far. And it's superior to other compounds discovered. And then it's we are at such times that we would really need kind of a beam of hope, and that is the picture I took a couple of years ago at the cottage on Lake Huron, and it shows us that there are spaces where sun could shine, and in that respect we could focus on specific targets, we could focus on specific projects and show some results. And again, in chemical space, it's like here, you could beam only on a small number of compounds, but it should be very focused, and you should rationalize why you are in that chemical space and what you want to discover there. And just the last slide, I would like to thank you, my colleagues as molecular design department, organic chemistry group, biological group, and really huge thanks to our main collaborator and the driving force behind CK2 project. It's Institute of Molecular Biology and Genetics in the Ukraine. And obviously, I would welcome any questions which you might have after this presentation.